Hello everyone, my name is Dr. Deepesh Neelam. I am an assistant professor at Department of Microbiology at JECRC University, Jaipur. Today I will talk on the nothing is impossible for the extremophilic microorganisms. So before I am going to discuss in detail, we have to know about what are the extremophiles. So extremophiles are the remarkable microorganisms that are thrive in the harsh environmental conditions which are present on the earth. The word extremophile is originated from the Greek word that means extremus plus philia, means those who loves the extreme environment. Later on, the term is coined by the Macaulay in 1974, and it is also known that the most of the extremophiles are archaea, but can also include the bacteria and eukaryota. But there is a question is arises why we have need of these extremophiles. So according to the astrobiology, extremophiles are those microorganisms which are able to grow at the higher extreme conditions. So on the basis of these extremophiles considered as a model organisms when they're exploring the existence of the extraterrestrial life out of the earth. Second thing is only the limited research studies have been conducted on them, so we have to know about more for the unexplored diversity by using the various culturable or unculturable techniques. Then the extremophiles can be a valuable resources for the novel biotechnological and biomedical products due to their biosynthetic properties. For example, mesophilic enzymes are not well suited for the harsh reaction conditions that required in the industrial processes because the lack of enzyme stability at the various conditions like higher temperature, lower temperature, higher salinity, and high acidic conditions. So at this stage, the extremophiles bioactive products could be the potential source to fulfill such requirements. So here it is the question is arises what kind of the extreme conditions are there where the extremophiles can be grow. So there is the some extremophilic conditions are present on there the extremophiles can be grow. For example, like the higher temperature, lower temperature, high saline condition, high alkaline condition, acidic conditions, pressure, and the other some specific dry conditions like heavy metals and concentration and the UV radiations. So such kind of things are also found in the entire world. For example, the Yellowstone National Park or Thar Desert is especially known for their high temperature. And if you are talking about the lower temperature, there will be found in the glaciers and the oceans. Similarly, the saline and alkaline condition will be found in the Sambar Lake, Great Salt Lake, and Bonar Lake. So based on these extreme conditions, in this my presentation, I will discuss only the important classes of the extremophiles. For example, number one is the cyclophiles. Cyclophiles are those microorganisms which are cold loving and growing optimally at the 15 degrees Celsius, and their growth range is -2 to 20 degrees Celsius. And there is some cyclotolerant like pseudomonas which are able to grow at 4 to 35 degrees Celsius. And if we are talking about their habitat, these cyclophiles are grow they at the polar oceans, glaciers, Antarctic soils, and the marine environment, etc. But here is how they can survive. There is just some mechanism behind that. On the basis of them, they can survive in this such kind of lower conditions, which are anti-freezing proteins, cold stalk proteins, membrane fluidity by the unsaturated fatty acid, and some exopolysaccharides. And if you are talking about the some examples like the Cyclobacter aquaticus, Pseudomonas antarctica, and Locaria polaris, these are the examples. There is the another class is acidophiles. Microbes, those are living and they grow at the very lower condition of the pH, like 1 to 5 pH. That is the very extreme acidic condition for the survival of any other microorganisms. So such kind of microbes are known as the acidophiles, which are able to grow at the acidic conditions. And where these conditions we can find out in the such like the acidic river, acidic hot springs, and the various mines, there is the sulfur compound common, etc. And if you are talking about their adaptation, the sugar coating and the higher content of the glycoproteins help to maintain their high relative internal pH during the survival in acidic environment. And if you are talking about the examples, sulfolobus acidochloridius and the hydrogenobaculum acidophilum are the some examples of these extremophiles. And the another type of the extremophile class is halophiles. Halophiles means salt-loving microorganisms which are able to grow at 0.2 to 5 molar NaCl concentration. That means 1 to 30% NaCl concentration. That is the very extreme conditions. There is the nothing is the another life is can grow. So such kind of habitat are found in the hypersaline conditions like the Great Salt Lake, Dead Sea, Sambar Lake, etc. And what mechanism they follows for their growth? That is the salt in and salt out process. And second one is the organic compatible solutes. So there is some these kind of conditions they maintain by the some organic solutes which are like glycol, pyamol, etc. These are the organic content which maintain their higher concentration of KCl ions by using the organic compounds. And if you are talking about the some examples like the halobacterium, halobrex, volcani, chlorobium, imilicola, and disulfovibrio halophilus are the very important examples of the halophilic bacterial classes. There is the another class that is the alkylophiles. Alkylophiles are those microorganisms which are able to grow at the higher pH. Means most of the alkylophiles are able to grow at the 9 pH, but there is some extreme alkylophiles also present there. These are generally grow at the 10 pH or above. And these microorganisms are found in the alkaline environment like the Mono Lake, Lunar Crater Soda Lake, and Lake of Magdigi, etc. And if you are talking about their adaptation, they can maintain their low cytoplasmic pH than the external pH value by the coordination of hydrogen ion and sodium ion pumps in their cell. So these are some unique characteristics of these halophilic classes and the other microorganisms which are able to survive in such kind of extreme conditions. And if you are talking about the example, the Rhodospira, Halochloris, Netornomonas species, and the Thiohalospira and Alkylophylla are some examples of alkylophilic microorganisms. There is another classes of the extremophiles like barophiles. Barophiles means those microorganisms which are able to survive at the higher pressure. At the that stage, there is the normal microbial cells cannot be survived. They immediately rupture at that atmospheric pressure, that is the 100 to 700 atm. So such kind of habitat where we can find deep in the earth and deep sea hydrothermal vents. These are some specific habitats where we can find these barophilic microorganisms. And if you are talking about the adaptation, the lipid unsaturated fatty acids like PUFA, the protein coat at the outer cell membrane proteins, enzymes, and the pressure controlling genes are responsible for their survival at the higher pressure conditions. And if you are talking about their examples, the Proteobacteria gamma and Halomonas ciliaria are the examples. There is another important class is radioresistor. Radioresistors are those microorganisms which are able to survive or they can tolerate the higher radiation at the various environments. For example, if you are talking about the UV radiation, there is one important bacteria is present that is known as the Deinococcus radiodurans, which is also known as the hardest bacteria which can be survived at the higher UV radiation concentration or higher UV radiation. So here is the point is how this bacteria can be survived at the higher UV radiation condition. So there is some DNA repair enzymes are present like the REC-A protein and which are able to matches the broken pieces of the DNA and splices them back together in the few hours. So at this stage, I want to tell you that there is another important class is present, that is the thermophiles. Thermophiles are those microorganisms which are able to grow at the higher temperature, and such kind of thermophilic bacterial class is not only important for the research purposes, but also these are important for the industrial purposes in the various industries like in the food industry, dairy industry, and the leather industry, and the enzyme industries. So what are the specific habitats where thermophilic bacteria can be found? These are hot springs, deep sea thermal vents, desert, compost, manure, and oil wells and the laundry, etc. These are the such habitats. There we can easily find out the thermophilic microorganisms. So here we have to know about that what was the first bacteria who was isolated in the first time. So Thomas Brock and their colleagues in the late 1960s discovered the first extremophilic bacteria. This is capable at survival at the 70 degrees Celsius, which is isolated from the Yellowstone volcanic hot spring that is known as the Thermos aquaticus. And naturally, it is a source of tag polymerase enzyme, which is used in the polymerase chain reaction for the amplification of DNA. Then the Kesafi and Lovely in 2003, they isolated the Archaeobacterial strand that is 1 to 1, that is 1 to 1, which is known as Geogamma brosi, and isolated from the Mothara hydrothermal vent. This strand is able to survive at the 121 degree Celsius. So these thermophilic bacteria further classified into the three types. Number one is the moderate thermophilic bacteria, extremophilic thermophilic bacteria, and the hyperthermophilic bacteria. Based on their survival temperature, so moderate thermophilic bacteria are able to survive at the 50 to 60 degrees Celsius, and the extremophilic bacteria are able to survive at the 65 to 80 degrees Celsius, and hyperthermophilic bacteria are able to survive 100 to 122 degrees Celsius. And for if we are talking about the examples like the Bacillus coagulans, Geobacillus stereothermophilus, Clostridium thermoaceticum, and Thermus disulfovibrio, Thermoanaerobacter, and Sulfolobas, Thermovoccus. These are the very important examples of the thermophilic bacteria. So now here is the question arises, how these thermophilic bacterial different classes can survive in the higher temperature, because this is the very important class. So there is the various molecules are responsible for their survival in the higher conditions of the temperature. For example, the mechanism, if we are talking about the mechanism which is responsible for their adaptation in the higher temperature, the membrane lipid have a ether linkage. That means more branched and more stable. So there is the higher molecular weight. So these characteristics increase the melting temperature of the membrane lipids at the higher temperature conditions. Second is the heat shock proteins. More hydrophobic interior prevent the unfolding or denaturation of DNA during the higher temperature. And another important point is the high guanine and cytosine content in the nucleic acid structure. And there is the another important point is the reverse gyrase enzymes. These enzymes help in the catalysis the positive supercoiling of the closed circular DNA, means the positive supercoiled DNA appear to the resistance degradation more than the negatively supercoiled DNA. So there is the very important function of the reverse gyrase enzymes to to catalyze the supercoil coiling of the DNA at the stage of the supercoiling. So there is the another thing is very important by the side by side the reverse gyrase, that is the DNA binding histone proteins or histone like proteins which are able to make the strong binding in the DNA molecules. And there is the another some potassium and magnesium ions we are working for the higher temperature in archaea protect the standard DNA from the phosphodiester bond degradation. So based on this adaptation or based on these conditions, we know that the in the extremophilic classes of the various extremophiles, thermophilic microorganisms are more diverse and they are more attractive in the field of research as well as in the industries. So here I will tell you about why the extremophilic microorganisms are more emphasized for their enzymes. Generally, extremogens are those enzymes which are produced by the extremophilic microorganisms. So here we can say that the discovery of new thermophilic microorganisms and their enzymes has a great impact on the field of biocatalysis, because the extremogens from the extremophilic microorganisms are more active and more stable at the higher and the various different stress conditions. And These extremogens are able to replace the uses of chemicals from the various industries of the environment, and they are the environment friendly and sustainable for the new generation of industries. In particularly in the extremogens of the various classes, thermophilic microorganisms have found the most practical commercial uses to date because their overall inherent stability at the higher temperature, as well as they have some another characteristics like they are also able to grow at the higher pH. So they have a multiple characteristics to survive and able to produce the some enzymes. Those are working at the not only for a single specific parameter, but also they are able to stable at the multiple parameters. So here is the in this table you can see that there is the various extremophilic bacterial classes which are able to produce various enzymes. If we are talking about the cyclophiles, acidophiles, alkalophiles, halophiles, and pyrophiles or pisophiles, these are able to produce the various enzymes. But if you will look at the end, there is some thermophilic microorganisms. These are able to produce more than 4 or 5 enzymes. So this class is very, very important in the industrial purposes, in the various industries like the food industry, like the dairy industry, like in the detergent industry and the starch industry, and the pharmaceutical companies, or for the medical purposes and for the removal of toxic compounds. So here is the important thing is why the extremophilic microorganisms are more important for the extremogens, because thermophilic extremophiles have attracted the more attention, in particularly for the extremophilic enzymes such as protease, lipase, cellulase, chitinase, and amylase have found their way into the industrial applications, as I have told you in my earlier slide. Then there is the another reason these thermophilic bacteria are have some reasons to stability at the higher temperature, but they have some more reasons like they have to avoid the contamination leading to understand the complications to reduce at the higher temperature. So these are very important class of the extremophiles, which is not only for able to produce the thermophilic enzymes, but also there is the another microbial life cannot be exist, or they cannot be able to do the some activity at the enzyme level. So the usage of such enzymes in the maximizing reaction accomplished in the food and paper industry, detergent industry, drug, toxic waste, and removal and drilling of the oil is being studied extensively. So based on these things, in my PhD work, I have did my PhD work under the supervision of Dr. Pawan Kumar Dadi sir and Dr. Akhil Agarwal sir. And during my study PhD research work, we found that various thermophilic microorganisms which are able to produce not only a single extremophilic enzyme, but also they are produced the maximum various industrial important enzymes. Those are capable to work at the industrial level. In this figure, you can in this slide, you can see there is the one stent that is able to produce the alpha amylase, which is used in the starch industry. And if you can see in the B figure, there is the protease enzyme producing thermophilic bacteria. Those are uses in the detergent industries and the protein industries. And if you can see, there is the C in the C figure, there is, you can easily observe the hydrolysis zone surrounding the bacterial strain. That means these are able to produce the cellulase enzyme. And if you can see in the D figure, there is the esterase enzyme, which is produced by the thermophilic bacterial strain to segregate the some content related by the twin AT. And in the figure D, you can see there is clearly the red color zone indicate the activity of L-asparginase enzyme. And at the end, there is the in F figure, you can see there is the anurelinase enzyme production by the bacterial thermophilic strain. Here, I will not disclose the name of strains because this data is unpublished. So I am not able to represent the name of these thermophilic microorganisms. Now, there is the these are the some qualitative analysis of the extremophilic enzymes. But in the industries, I want to tell you that these are not required the 100% purification of enzymes. Generally, in the industries, they only want the semi partial purification of the enzymes. But in the some cases, if we are talking about for the pharmaceutical industries, or for example in the medicines, or if we are talking about the particular any research, there is a need of the purification of enzymes, but industry doesn't want to uh, waste their time for the complete purification of these enzymes. So on the basis of based on this research work, here I will close my this presentation, and I'm very happy to tell you that you are now at this stage. You can know about what are the extremophiles and where they are live and how they can survive, and extremogens, and you know very well now about the extremogens, how they are great economic potential in the many industrial processes, including the food, agriculture, and chemical and the pharmaceutical applications. But still there is we have more need to understand about the extremophiles, because till to date only the less than 1% of the studies has been done in the various extreme conditions for the extremophilic microorganisms. If we are talking about the some metagenomic work, through that we can identify there is the various microorganisms are present in that particular natural sample. Those are able to show the genes regarding the extremogens, but there is some lack of cultivation techniques present till to date we have. On the basis of them, we cannot able to grow these microorganisms into the in vitro conditions. But now I can say only one thing: in the upcoming future time, we can able to grow these such kind of microorganisms into our lab. So with this slide, I will stop my content regarding the extremophilic microorganisms and their applications. So this is the Department of Microbiology, Dr. Varsha Gupta, Associate Professor and Head of the Department in our JECRC University, and Ms. Devki Sen and Dr. Ravi Kant Rai are my colleagues. And here is some references which I have added here to know where I have used this content for my slides. Thank you very much for listening me patiently. If you have any query, please write in the text box. Thank you.
Thank you to Deepesh Neelam, Assistant Professor of the Department of Microbiology at JECRC University in India, for submitting that pre-recorded talk.